Hello, everyone, and it's my pleasure to welcome you to today's session. My name is Doru Florescu. I'm with Park Systems, and I'm one of the organizers of our workshop series. Today, we present to you the Q3 calendar year 20 session. We have two very exciting talks by presenters that I will be introducing next. And again, I wanted to remind you that this series is a quarterly series. We will provide details on the time and date for our Q4 event and who the speakers will be in the last quarter of this year. Coming back to today's session, it's my pleasure to introduce Dr. Sung Park and Dr. Matthew Young, the two presenters. Dr. Sung Park is the CEO of Molecular Vista. He has a Ph.D. in applied physics from Stanford University and over 25 years of experience in the STM AFM field. I know Sung for a very, very long time, and I'm very happy that he can join us today. And Dr. Matthew Young is one of the Park Systems staff. He leads the US Applications and Service team. He has a Ph.D. from Kansas State University, and he will be presenting some applications that we have in line with our automated AFMs. With that, just to remind you of the format, the speakers will make their presentations. We can take a few questions at the end. And then if you have any questions that you would like to submit, please do so via chat. We'll take these and follow up with you guys offline. So let's get going. Thank you very much.
Thank you, Doru, for the nice introduction and for this opportunity to speak in this webinar. The title of my talk is Atomicscale AFM-based IR's Hyperspectral Analysis for Advanced Manufacturing. Really, the atomic portion will be more limited to the vertical direction, so the height of the film, and more nanoscale in the lateral direction because we do not have true atomic resolution. There are many microscopy and microanalysis techniques currently available for monitoring advanced manufacturing processes. Well known, of course, is the electron microscopy, with which you can actually get very nice elemental information. With high-end microscopes, you can actually get atomic resolution. But even so, it's very difficult to assess the molecular composition with this type of tools, even when you have the elemental information. Also very commonly used is scanning probe microscopy with atomic force microscopy or AFM, along with other techniques such as mechanical mapping, where one can analyze the topography and indirectly the presence of different components, for example, by measuring mechanical mapping properties. So here, for example, is a kind of a ternary compound. It's a pharmaceutical compound that consists of PBMA, PLMA, two different polymers with a dexamethasone, which is a steroid. Here you can actually get a pretty nice image of where the different components may be at this sample, but really it's very hard to tell where things are. Even if you know the bulk properties, it would be very hard to, with confidence, identify where different components are going. In order to actually identify and get molecular information, a tool such as FTIR, where you can actually measure the infrared absorption of different bonds and get a chemical fingerprint, is quite widely used. However, because in this case the wavelength used is on the order of microns, the spatial mapping resolution is also limited to on the order of 5 microns. So for a long time, people have been wanting to get a chemical signature of a nano-sized particles materials. And so people have been looking into ways to combine an AFM with these optical spectroscopy techniques. And photo-induced force microscopy, which I will be talking about today, which I will be just referring to as PIFM, IR PIFM, is where you combine IR spectroscopy with an AFM. So what we do here is we use two mechanical resonances of the cantilever, AFM cantilever, as two independent force sensors. Instead of using the F0 shown in the far right, we use the more stiffer resonance mode, F1, to do the AFM feedback control. We do so that we can actually get very close to the sample surface, so that when we excite the sample with optical beam, that photo-induced force can be used to excite the first mode, F0 mode of the cantilever. So what we do is when we use the F1 mode to feedback control the AFM topography, we excite the laser, in this case a quantum cascade laser, QCL. We modulate it at a frequency FM. When the laser turns on and off at FM, that photo-induced force will be also modulated at the same frequency. So you would have this cosine of 2 pi FMT dependence. Now, at the same time, as I mentioned, the tip is going close and farther away periodically at this frequency F1. Since the dipole force, which is kind of the nature of this photo-induced force, dipolar, is a very gap-dependent force. Basically, when it's closer, the force will be stronger, and farther away, it will be weaker. So it will also have this sinusoidal cosine of 2 pi f1t behavior. So when you have these two cosine terms, I'm sorry, go back, you will create two sidebands, one at the sum frequency and one at the difference frequency. We actually choose the FM so that it's at a difference frequency of the F1 minus F0, so that this one of the sidebands actually fall right on top of F0, and that will excite the resonance of F0. So by monitoring the amplitude of the first mechanical mode, F0, we can see how strong the optically induced force is. So here you can see by using two lock-in amplifiers, we can actually acquire the standard AFM images as well as this PIFM image at the same time. So if we take this example of a PS-LDPE blend, you can, in the middle panel, you can see that we can get topography and phase, which is kind of a mechanical elasticity mapping from the standard AFM mode. And then if we at the same time fix the laser to 1466, which corresponds to a vibrational mode associated with LDPE, whenever tip is on top of that LDPE molecule, there will be more attractive force, and that F0 amplitude will increase. So by monitoring that amplitude and plotting it out as a function of location, we can generate that red image. And there, what that means is wherever you see a strong attractive force or signal, that's where the LDPE molecules are. Without changing the location, now we can change the wave number to 1493, which will map out the polystyrene molecules. So you can create that map and then combine them, green and the color map together, to create a chemical map image. So that way we can actually get both the topography as well as the chemical information. The other way we also operate the tool is if you see a feature you would like to see the chemical signature, you can go there, just like when you do a other type of spectroscopy and AFM. and then you can actually sweep the laser while you're monitoring the amplitude of the F0 or resonance. Then you can actually generate a PFM spectrum, which in many cases will correlate with the FTIR spectra. So we'll use this sample as an example. Here is a PLA-ACM composite film. And since we know the two components that make up this film, we can actually use the two bands associated with those components, PLA and ACM. So PLA has a peak at 1750 and ACM at 1720. We can generate those blue and green images. At the same time, topography is acquired. But then we noticed that at 1720, there is some dark spots within the grains or islands of ACM. And when you see such a oddity, you can zoom into it. So we can zoom into that white square box, and it's shown at the left bottom image. You can see that there are these inclusions in the ACM islands. And in such cases, you can actually go to those three spots. We show the black dots are where we take three different spectra. And those spectra are shown on the right. And we can see the blue spectra associated with the PLA, the green spectra associated with the ACM, and then the red spectra associated with those inclusions. And we can see that that inclusion has a very strong and unique peak at 1072. Then you can actually go there and take another image at that wave number to create a full composite image. So that's what we're seeing at the bottom left, where we show the green to represent the ACM molecules, blue for polylactic acid molecules, and then the third component, which we don't know what it is, but it is characterized by that wave number. So when you compare the spectrum that we just acquired, even though they were acquired from a very local region, you can see that it agrees very well with the published FTIR spectrum. So this kind of gives you a quick overview of how one might use a PFM instrument in conjunction with AFM. So now we can briefly revisit this sample where there was this ternary pharmaceutical compound, composite material. We saw that adhesion and the stiffness images really highlighted kind of complementary images. But since we know the peaks associated, vibrational bands associated with PBMA, PLMA, and dexamethasone, we can actually image this same sample with such a wave numbers. We can see that PLMA at 1729 wave number and PBMA at 1468, they actually show features very similar to the stiffness and adhesion. But in addition, we can see that dexamethasone obtained at 1666, we can see that it occupies only the small islands that PBMA was highlighting. So then by combining all three of them, you can see with confidence where the different chemical components are going and can augment the AFM and the nanomechanical data they were able to get. Okay, now that we've gone through just the basic operation of the tool, let's just explore some applications in advanced manufacturing. As you know, defect analysis is a very important element of advanced manufacturing, because if something that you did not expect to show up in the process, or after the failure, you find something has been there, you want to know what they are so you can figure out where the defects might have originated from. So here, because PIFM has that nanoscale capability, a spatial resolution, and the very good sensitivity, we have here a defect that is only about 100 nanometers in lateral dimension and only about less than 5 nanometers. You can see the full Z scale on the right in circle, the red circle. It's less than 5 nanometers thick. And you can go to on top of the particle to generate the blue spectrum, and then you can go to the substrate to generate the purple spectrum. And it turns out that this particle matches the Teflon peaks very well, giving a very clear indication this particle is a Teflon particle. Now, we can do the same thing with inorganic materials as well. Here we have a particle that is only about 18 nanometers, probably a little less than that, 15 nanometers in thickness. Extent laterally, it looks like it's 50 nanometers, but as you know, with AFM, the AFM's tip width will convolute the particle. So probably this particle is on the order of about 20 nanometers wide and similar thickness. And we can see that when we take the spectrum on top of the particle, the peak associated with that is on the order of about 1100 wave numbers, whereas the substrate has a peak around 1060. The 1060 is a peak associated with amorphous silicon oxide, so it's probably showing the native silicon dioxide because the substrate was a silicon substrate. But the little particle, 20 nanometer particle, has a signature more associated with crystalline silicon dioxide. So we can probably know that it's a silica particle that is being observed on a silicon substrate. So as you can see, you can do very good analysis of very, very small defects, both organic and inorganic materials with PIFM. When you don't know when the peaks are a little bit more complex and you have to rely on a online library, you can actually feed in a spectrum from PIFM into a library to look for a match. In this case, we actually took a spectrum on a known piece of polyvinylidene fluoride and generated that red spectrum. And then we fed that into a online FTIR library. And the very first match with the highest quality index is indeed the same material. So you can tell that you can use PIFM spectra to do defect analysis. The other next topic will be how many processes in advanced manufacturing utilize multi-layers of material, both organic materials as well as inorganic materials. So analyzing how they interact with each other and what the actual thickness is compared to the intended thickness, and those kind of things can be done with PIFM very nicely. So here is an example where we're looking at two polymers. One is a sulfur-containing polymer in yellow, which is about 15 microns in thickness. And then it's capped off with a polymer we will call P-delta. And that's on the order of 50 to 100 nanometers. And the question is the interface very, very sharp, or is it kind of interacting and mixed, creating a very solid integrated interface? So here you can see that when we put that into a epoxy and you microtome it, you can actually now expose the cross-section and we can actually analyze it. In the middle, we see the topography where it's very hard to tell what components are where and what the interfaces are. But when you actually take PIFM image at 3 wave numbers, at 1512 for the epoxy, and 1734 for the PDAGDA, and 1679 for the sulfur containing, I'm sorry, I don't know why it's advancing by itself, sulfur containing SDIB, and create a composite image, you have that image in the middle. And you can see that the interface between the epoxy and the PDAGDA is very sharp, the green to blue, but the blue to red interface is somewhat has a transition in terms of grayscale. And you can actually take 30 spectrum across that interface, 1 through 30, with 10 nanometer spacing for each spectrum, and shown on the left. You can see by looking at the peak at 1734 that there is a very sudden jump in the peak height from 5 to number 6. That's the interface between the epoxy and the PDAGDA. But on the other hand, the same peak actually gradually decreases and approaches a saturate kind of steady state height from 13 to about 20. So there is a gradual decrease indicating a mixing. And here we're actually comparing the ratio of the peak at A, B, and C divided by the peak at A, and you can see that the red corresponding to the SDIB and the blue that corresponds to PDAGDA has a gradually mixed from about 120 to 190 nanometers. So actually there is about 70 nanometer of mixing instead of a sharp interface. Here we're actually seeing a cross-section of a inorganic material. It's a silicon piece that was just cleaved, and we can see that looking at this material 1, which is based on this oxide peak at around 1100, we can see a difference looking at the PIFM image at the bottom left, that there is a difference between the oxide near the surface and as we go into the surface. And you can actually see material 2 in red, and you can actually use multiple spectra to see the transition between the material 1 and material 2 to study the interface chemically very, very carefully. Okay, the other is a analyzing monolayer film. Monolayer films are used widely both in semiconductor industry and in biomedical and perhaps other manufacturing environments as well. So we wanted to show that application. Here we're looking at a self-assembled monolayer of octadecyl trichlorosilane on top of silicon oxide. The sample substrate is grating, silicon oxide grating, that's covered with a 30 nanometer of silicon nitride and 5 nanometer of silicon oxide. And then on top of that 5 nanometer of oxide, the ODTS self-assembled monolayer is formed. And when we actually take the spectrum across the structure, here in the middle it shows 1 through 18, and we can assemble the spectra acquired at the top surface and at the bottom surface. And you can see that there is very good repeatability among the different surfaces, upper and the lower surfaces. And you can see that lower surface has a much stronger peak at 2917 compared to the upper surface, showing that at the lower surface, we actually have tighter, more dense packing of these SAM molecules. And we can attribute that actually to the difference in the nature of the silicon oxide. We actually have a recorded webinar on this topic, so you can actually go visit to learn more about what the nature of the oxide was in that link, using that link. But it shows that we can actually see monolayer films very, very nicely. This is another example where two different types of silane molecules are used to define an area where DNA molecules are attached. One of them has is fluorinated so that it prevents the deposition of the DNA molecules. The other one does. So here you can see from the topography image that really it's very, very thin sample, and that by using two wave numbers associated with the two different type of silane, we can highlight the silane very nicely, the two different silane films very nicely. You can tell that the boundary is actually neither. It is a different material, and that's why it doesn't get highlighted in either images. Just shows that PFM can have excellent sensitivity to work with monolayer films. The other is, of course, to visualizing distribution of components at nanoscale. So here we show a laser toner particle, which has actually multiple components. When you take a FTIR of such a sample, as shown here in red, you don't expect the nanoscale PFM spectra to correlate as you would expect on a homogeneous sample, because in nanoscale there would be inhomogeneity. So here we can see that by looking at three different locations of the grains, you actually have different contributions. The yellow contributes strongly at 1100, the dark blue at 840, the light blue around kind of the broad range of 1400 to 1500. So you can tell that FTIR for inhomogeneous sample will be a ensemble or sum of many, many local PFM spectrum. Now, based on these type of information, though, we can create a chemical map, because here we can take an image at 1100, 1454, and 1513 wave numbers associated with different components that you are creating this laser toner particle, and you can see how they are distributed in the sample. Here, another example of a kind of how different components are distributed. This is a polymer-polymer blend solar cell. It has a donor and acceptor components. And by looking at comparing the PFM spectra and the FTIR, which has good match, we can see that at 1453, we would be able to highlight the donor molecules. And at 1706, you would be able to highlight the acceptor molecules. The topography and the phase really do not give much information. But if you take the image of 1453 for the donor and 1706 for the acceptor, you can combine them and to create a very nice chemical map. And the reason why it works very well is this works well as a solar cell is because it's well integrated. And you can see, you can then do a grain analysis to figure out what is the size associated with this material for different amounts of additive. And when you do that, here you can actually get a rough idea of the grain sizes by doing resonant soft X-ray scattering, which is a very widely used technique. You can actually get a sense of what the grains are. Then you can compare the grain size inferred or calculated from PFM versus this X-ray scattering. and then plot it against actual device characteristics. And you can see in the B where they are looking at the inverse of the device short circuit current versus the domain width calculated via PFM corresponds much better with the device short circuit current shown in red. The same thing for photoactive volume shown in C when it's calculated plotted against the PFM domain size. It agrees much better than the soft X-ray scattering curves. So we can see how PFM can be used to define the domain sizes of blended samples. And then what we can close with an example is a visible absorption. We've talked about IR absorption to do chemical mapping, but in this case, it's a perovskite photovoltaic film, whereby applying a bias, you can actually change the absorption profile of the film. So here, going from 0 volt to -1 volt, when you apply that voltage shown in the cartoon A, you actually pull the cations toward the top. When the cations move toward the top, the absorption at 650 nanometer increases quite a bit. So you can see much more widespread absorption compared to 0 voltage. Now, as you then gradually lower it back to, or increase it back to 0 voltage, you see the cations moving back away from the top electrode, and you actually see a decreasing amount of absorption. And then now, as you apply bias voltage all the way back up to 1 volt to counter the original -1 volt, you can see that the perovskite film has reverted back to the original ionic concentration, so that now you have a situation that is very similar to the original 0 volt. Basically, they're trying to use this type of photovoltaic film for dynamically controlled photo detectors, so that it can be used along with kind of artificial intelligence. Depending on the different conditions, they will adapt the absorption profile to see things better. So here, basically, PFM is used to measure the visible absorption in addition to the chemical analysis you can do with the IR wavelengths. So I'll finish by just sharing that the technique complements other analytical tools. As we mentioned, the elemental information can be acquired by electron microscopes with very high resolution. But molecular information can be best acquired prior to PFM via nanosims, top sims, to about 200 nanometers. But with PFM, we can get down to about 5 nanometers in spatial resolution and can complement the other techniques very well. So in summary, PFM provides nanoscale IR and visible absorption analysis along with all the standard AFM modes. It currently offers 5 nanometer spatial resolution and monolayer sensitivity, works very well with both organic and inorganic materials. It complements other high-resolution elemental analysis techniques, providing molecular information, and applications for various facets of advanced manufacturing have been shared. And PFM is really can be applied in diverse applications, and many additional examples from biology, geochemistry, 1D, 2D materials, and plasmonics and photonics and other fields can be found in our website. And I'll just end with my our email and website contacts. That does it for me. Thank you.
All right, thank you very much, Dr. Park. Hello, everyone, and thank you very much for your attendance. I'm Matthew Young. My discussion today is automated AFM for metrology applications in semiconductor manufacturing. So to begin with, for the benefit of those of you who may not have seen our previous webinar or maybe new to Park Systems, Park Systems is a fully integrated atomic force microscopy company. We were founded in 1997 by our CEO, Dr. Sang-Il Park. We are headquartered in Suwon, South Korea, in the Korea Nano Fab Center, shown in the picture at right. Since 2015's IPO, our revenue has doubled in that time. Park Systems employs approximately 250 employees worldwide. We have over 1,750 tools installed, including over 160 tools in industrial environments in various fabs across the world. And we hold 32 patents, which speaks to our technical prowess in the field of AFM. To further demonstrate our global presence, we have 10 regional offices in 9 countries around the world, located in the Americas, East and Southeast Asia, and Europe, as well as 28 distributors in major countries around the world. Our AFM line includes a variety of research and industrial tools for various sample types and environments. The research line includes tabletop tools as well as high vacuum environment tools for a variety of applications with a number of options for measurements of material science, electrochemistry, and the life sciences. We also have a line of industrial tools for primarily 150, 200, and 300 millimeter diameter wafer applications with a strength for non-destructive inline analysis as well as high repeatability. Now, across this entire product line, the main backbone is the decoupled X, Y, and Z scanner, which affords great repeatability across tools and nanoscale precision for, again, a great variety of applications. Now, as I begin the discussion of applications, I want to keep bear in mind that these are primarily focused upon semiconductor applications in a manufacturing environment, but that many of these can be applied to various fields, not just semiconductor, but even the ones shown here, as well as any particular sample type, not simply wafers. So to begin with, starting out very simply in the applications, I want to discuss surface roughness. Now, the challenge addressed with surface roughness is detecting out-of-control wafer process steps in an inline non-destructive measurement. So AFM steps in here. We can perform surface roughness measurements at representative measurement sites across the wafer and begin to get a picture of if there is a particular variance that may be caused, for example, by a CMP polish step, a deposition step that has gone awry, anything that might show up in the roughness measurement itself. And by performing these measurements, we can either have a localized assessment of the quality of the wafer at that process step, or even generate a full wafer contour map, heat map of the roughness as shown here at the right. Now, this application is good for when you have what you seem to be a very distributed roughness histogram. However, for the case in which you have a discrete feature, a defect, let's say, on your wafer, for this, there is a further application called Automatic Defect Review, ADR. The challenge here is for wafer defect root cause analysis, it requires very accurate size and shape data. So this is where ADR steps in with nanoscale precision. The basic workflow of this method is an optical inspection tool inline in the fab generates a defect map, and then that defect map is sent to the Park AFM. The Park AFM aligns its own coordinate system with that of the optical inspection tool, and then will image as many or as few of the defects in that map as necessary. The defect shown here is quite large, about 743 nanometers in size, but we can nanometer as small as even, say, 10 nanometers if required. Just depends on the particular application. So this is a very good demonstration of the kind of quality assurance that the AFM can be used for in an inline environment. Moving forward, another particular challenge is using a measurement analysis technique that allows for length scales beyond the typical scale of AFM, 100 micron, while still keeping the accuracy, particularly in the Z direction, that an AFM affords. So for this, we use a long-range profiler, which again, it couples the Z scanner accuracy with now a 50 millimeter scan range to make use of AFM for particular applications that could be encountered in a variety of different measurement circumstances. So just for an example here, I give a 1 millimeter scan to show a calculation of height range as well as pitch, pretty commonly used for just an assessment of your feature dimensions in a manufacturing environment. This can also be used and is typically used for CMP applications in which you would assess the oxide and metal layer erosion during a CMP process. Segueing from the long-range profile, I want to discuss wafer edge metrology. And the challenge here is to increase the yield of a particular product on a wafer by minimizing what's called edge exclusion. Just for an understanding of what edge exclusion means, I want you to consider the following example: 2 identical 300 millimeter wafers, upon which a various series of processes will be run to develop architecture with a die size of 12 millimeter by 10 millimeter, which is fairly standard for memory product applications. The difference between these two particular wafers is the one on the left has an edge exclusion of 4.5 millimeter, while the one on the right is 2.0 millimeter. Now, while at first that might not seem like a great deal of difference, the one on the right with the smaller edge exclusion does have 22 additional dies on its surface in full intact compared to the one on the left, representing a 4% increase. So this kind of increase, particularly for high manufacturing environments of memory products, is especially important to meet the demands, increasing demands of the memory product market. Now, the way AFM handles this particular metrology is to conduct long-range profile measurements across the edge of the wafer to measure the die architecture from which the edge exclusion can be calculated directly. I'll give an example here. In this case, we see that we have at the left side a clear die architecture. These features about 150 nanometers in height until we move along, sloping down until we reach the wafer edge. And by automatic calculation, we determine that the distance between the wafer edge and the die architecture is approximately 2.3 millimeter. And so this kind of measurement can be conducted, again, inline, non-destructive in a manufacturing environment, and can be done at various process steps to assess the quality and the integrity of the die at the wafer edge. A final point here is 2D pattern analysis, which is meeting the challenge of assessing dimensional variance in various 2D pattern features, such as through silica bias is one example. Interlayer connects in a memory product is another example. So for this, PARC-AFM allows for statistical analysis of those arrayed 2D features to assess the variance within that particular feature in a particular region. One example given here, in this image we detected 16 individual cells of a particular kind based on the desired height and the cell size. And here we have a full generated table of the height, range, and roughness with a standard deviation and range, the full statistics for each of these pads. And this analysis can be applied to a variety of features: 2D grading, a pad, a bump, as I show here, a pad, a pit style as another option. For customers who are working with charged coupled devices, microlens options are also available to detect the radius of curvature and the roughness of the microlens, as well as for LED manufacturers, a pattern sapphire substrate option to detect the height and the base width of the particular feature. To round out my discussion, I want to discuss 3 particular expanding applications of AFM. Starting with one challenge that is being driven by innovation in the memory sphere and the processor sphere for smaller nodes or co-optimization to decrease the feature length between existing nodes. This is being met by a new mode of scanning introduced by PARC Systems called narrow trench mode. And what differentiates narrow trench mode from normal AFM scanning is instead of the single line raster scan across the fast scan direction to generate the grid of data points to generate the final AFM image, narrow trench mode uses a discrete series of measurements to go point by point to collect each data point individually and thereby create the final image. And using this method, it does unlock the potential to measure particularly features that are narrower than could be measured by standard non-contact mode imaging. This image in particular shows 3 different probes with narrow trench mode or standard non-contact mode. And the red showing with the sharpest tip we had available, and narrow trench mode, we were able to measure features that otherwise would be unable to be resolved by traditional AFM, non-contact mode imaging, or would even be imperceptible to it altogether. Another expanding application is to take the metrology applications that we've discussed so far, primarily for wafer samples, and move those into samples that are large or heavy, beyond, say, 500 millimeter in length and 8 kilograms in mass. For this, Park has introduced the Park Systems NXTSH, or tip scanning head. The main innovation here is it takes the existing technology backbone of the decoupled X, Y, and Z scanner and combines it into a single self-contained unit. Now, this allows for the ability of the unit to scan a variety of very large samples, which can be in a tool that can be customized for different sample sizes and weights, while still keeping that same nanoscale precision. Finally, and conveniently rounding back to Dr. Park's presentation, another challenge is inline material composition analysis. And for this, we have worked with our partner, equity partner, Molecular Vista, of which Dr. Sung Park is a founder, to have a photo-induced force microscopy unit in addition to our existing AFM platform. And thus, that concludes my presentation. And if anyone has any questions, I'm happy to answer them here or offline if they come to you at a later time. Thank you very much for your attention.
Hi, everybody. I think we do have a question. I'm trying to see if I can read it here. So what is the Z height measurement range? And what's the max aspect ratio of the trench that can be imaged? So two questions, Matt.
Understood. Well, first question, the standard unit on our AFMs has a maximum range of 15 micron, but there is now recently introduced for both inline automation as well as research applications, a long range head, which has a range up to 30 micron, which is available for purchase. As for the second question, I can't really give an exact answer on that. This mode is still rather new, but we are pushing the boundaries certainly on this application. So I would say aspect ratio 3 quite comfortably, 4 quite possibly, but this is, again, evolving, so there are innovations all the time that's really pushing that aspect ratio further and further, along with the tip technology, which is helping to enable our narrow trench mode capability.
Thank you. So I just wanted to close our meeting up right now and remind everybody that we can follow up with questions offline. We will provide the recording link to you guys, and please feel free to contact the presenters directly with any additional questions that may occur in the future. So thank you again for joining in. Please stay tuned for details on our Q4 event coming up, probably towards early December. And we look forward to seeing you at the future session. Thanks, everybody. Goodbye.